Hello everyone, welcome to We Learn and welcome back to the class Molecular Basis of Inheritances. In the last class of Molecular Basis of Inheritances, we studied that DNA is basically made up of three parts and it is made up of a nitrogenous base, a phosphoric acid and a deoxyribose sugar. So if we talk about these three structure, that is a phosphoric acid, this is the phosphoric acid, this is deoxyribose sugar, this is the deoxyribose sugar, and this deoxyribose sugar do not contain a hydroxyl group at second carbon. This is the hydroxyl group at first time, but at the second carbon it do not contain any hydroxyl group. At third carbon it contain an hydroxyl group. At fourth carbon it contains CH2OH group. So this phosphoric acid and this hydroxyl group of this carbon atom, that is fifth carbon atom, combined with each other and release a water molecule and form a bond. The bond formed as a result of this will be known as phosphoester linkase. This is the phosphoester linkase. On the first carbon, this is the first carbon, C1, there is an hydroxyl group. And this hydroxyl group react with either the nitrogen group of purine or pyrimidine. Let us say this is the structure present, the double member ring structure. Double member ring structure means it is a purine. So this double member ring structure contain nitrogen at 1, 3, 7 and 9 position. The ninth position nitrogen atom combined with this hydroxyl group and form a linkase. This linkase is basically what we call CNC linkase. So this is the basic structure which form the polymer. As a result of this, if we talk about the further structure, the product of this reaction would be P double bond O, a hydroxyl group, again an hydroxyl group, and this is connected by O CH2, this CH2, and there is a release of two water molecules, of course. So there is a release of water molecules, and this is the CH2 of the deoxyribose sugar. And on the first carbon, there is again the nitrogenous base. In this case we assume this nitrogenous base to be purine. I am just drawing this basic skeleton structure of the purine. I am not completely drawing the structure of a purine. So basically this is the resultant product of this reaction. So if we talk about the bond formation, there are various bond formation in this deoxyribonucleotide structure. So this is the complete deoxyribonucleotide structure. So if we talk about the complete nucleotide structure, this is the carbon atom. This is the nitrogen, and again there will be a carbon atom. So this linkase here, this is what we call CNC linkase, and this linkase is basically a glycosidic linkase, and this linkase is from first carbon of the deoxyribose sugar to the ninth carbon of the purine. So this is with, in case of purine, it is first carbon to the ninth carbon, glycosidic linkage. So this is glycosidic linkage. But in case of pyrimidine, instead of, because there are only 6 member ring, instead of first carbon, there is first carbon to the first prime carbon of pyrimidine, glycosidic linkage. So this CNC linkage is 1' to 9' glycosidic linkage in case of purine, and in case of pyrimidine it is 1' glycosidic linkage. So this is glycosidic linkage. If we talk about this bond here is basically phosphoester bond. This bond here is basically what we call phosphoester bond. So this is basically phosphoester linkage. Phosphoester linkage. So basically this linkage is the phosphoester linkage and this linkage is CNC linkage. Further, this deoxyribonucleotide, this is the structure of this first deoxyribonucleotide. This deoxyribonucleotide structure form a long polymer. So basically these deoxyribonucleotides form a long polymer structure which is called as DNA. So DNA is basically the polymer of deoxyribonucleotide. How this structure is formed, we will discuss in this next detail. Now we will learn the structure of a DNA molecule. So let's start three-dimensional structure of DNA. If we talk about three-dimensional structure of DNA, the three-dimensional structure of the DNA was basically given by Watson and Crick. This three-dimensional structure of DNA, which was given by Watson and Crick, is basically based on two structures. This is basically based on Chargaff rule. This is basically based on Chargaff's rule, and it is based on Franklin and Wilkins structure. So basically Watson and Crick three-dimensional structure of DNA is basically the outcome of Chargaff rule and Franklin and Wilkins structure. What did Chargaff do? Chargaff did the chemical analysis of DNA. So this is the basic of three-dimensional structure, how the three-dimensional structure of Watson and Crick arised. So it arises from Chargaff rule plus Franklin and Wilkins structure. So we will study the Chargaff rule and Franklin and Wilkins structure of DNA. If we talk about chemical analysis, he did chemical analysis and he obtained that the amount of adenine is equal to thymine, or you can say the molar concentration of adenine is equal to thymine. Similarly, he gave another statement, that is the molar concentration of guanine is basically equal to cytosine. So their molar concentration of adenine is equal to thymine and guanine is equal to what? Cytosine. That means that is purine is equal to pyrimidine. In a structure, purine is equal to pyrimidine. That means the content of adenine plus guanine, is equal to thymine plus cytosine, or you can say in any organism which have DNA as the genetic material, purine upon pyrimidine because the structure amount is equal, so this ratio, that is purine upon pyrimidine ratio, will be equal to 1. Molar concentration of sugar. He further stated that the molar concentration of sugar is equal to molar concentration of phosphorus. But the ratio, again stated that the ratio of A plus T upon G plus C varies for different species. It varies for different species. This ratio A plus T upon G plus C, this ratio is basically known as base ratio. If we talk about the base ratio, this base ratio is, if we talk about prokaryotic cell, in prokaryotic cell, the G plus C amount, this prokaryotic DNA GC rich DNA, because the amount of GC is more. So A plus T upon G plus C जो ratio है, that should be equal to less than 1, that is less than 1. But in case of eukaryotic cell is 80 rich, because it is 80 rich. Normally the 80 rich means A plus T upon G plus C ratio is greater than 1. So this is the base ratio. The amount of purine is equal to pyrimidine. The purine and pyrimidine ratio is equal to 1. This is the base. This is equal to 1. This is the Chargaff rule. And the base ratio means A plus T upon G plus C का जो ratio है, that is not equal to 1. So base ratio is not equal to 1, and base ratio basically varies for different species. So this is the Chargaff rule. Chargaff basically did the chemical analysis of DNA and said that adenine का जो amount है, that is equal to thymine, and guanine amount is equal to cytosine. Therefore, the amount of purine is equal to pyrimidine. That means adenine plus guanine is equal to thymine plus cytosine. That means if we take the ratio adenine plus guanine upon thymine plus cytosine would be equal to 1, and this is true for all species. But if we talk about the base ratio, it varies for different species. It is different for the different species. In case of prokaryote, it is less than 1 because the prokaryotic DNA GC ratio is more. But in case of eukaryote, this is 80 rich DNA, and because of the 80 rich DNA, the amount of 80 rich DNA is more. That means this amount is more, this amount is less, therefore the value is greater than 1. So base ratio is clear. Next is the Franklin and Wilkins structure. They studied DNA with this technique X-ray diffraction. So basically they used X-ray diffraction technique. So they basically use the X-ray diffraction technique for realizing the structure of DNA, for solving the structure of DNA, and observed that DNA is basically a double helix. And basically they observed that DNA is double helix and its width is constant. So they already realized this part. They said that width is constant, and they also measured the width, that was 2 nanometer. That means the width of the DNA was 20 angstrom. And they also realized that in each turn of the DNA is basically 34 angstrom long. Each turn of the DNA is 34 angstrom long and approximately contain 10 bases. So basically, if we talk about this structure, they already realized, if we talk about the Franklin and Wilkins structure and Chargaff rule, they already realized that adenine amount is equal to thymine, the molar concentration of guanine is equal to cytosine, and the molar concentration of sugar is equal to phosphate. So this was derived from Chargaff. And we know that DNA is a double helical structure. DNA is a double helical structure. Its width is 20 nanometer and each turn of the DNA contain 10 bases, which is of around 34 angstrom or 3.4 nanometer. So this information was already there when Watson and Crick starting studied. Based upon this information, Watson and Crick gave three-dimensional structure of the DNA. Three-dimensional structure of DNA. So in the next class, we will exactly talk about what was the Watson and Crick structure, that is three-dimensional structure of DNA. So in next class, we will study the Watson and Crick three-dimensional structure of DNA. Thanks for watching the video. If you have any doubt regarding any chapter in year 12 or 11, please mention that in the comment section. I will reach out to the doubts and I will also start making videos on that part. Please hit like, subscribe and comment in the comment section. Thanks for watching the video and see you in the next video.